Good morning, everyone. Welcome to Grand Rounds. This morning we have a visiting professor to give our talk. His name is Jaakko Kaprio. He's a professor of genetic epidemiology at the University of Helsinki, Finland, and research professor in behavioral genetics at the National Institute of Health and Welfare in Helsinki since 2001. In 2012, he was appointed to a coveted Academy of Finland research professorship for 2013 to 2017. He has over 800 original scientific publications, and he was director of the Academy of Finland Center of Excellence in Complex Disease Genetics from 2007 to 2011. Dr. Kaprio has worked in epidemiology, in particular genetic epidemiology. His central interests have been in the study of adult chronic disease and the development of their behavioral risk factors with a focus on substance abuse and addictions. In particular, genetic and environmental factors affecting smoking and nicotine dependence have been studied using twin, family, and molecular genetic approaches. He has worked with the Finnish cohort studies since 1976 and with other population-based samples in Finland, and has engaged in extensive international collaborations with research in the US, Europe, and globally. He is currently an associate editor for nicotine and tobacco research and addiction, and serves on the editorial board of 5 other journals. He was president of the Society for Research on Nicotine and Tobacco European chapter in 2011 and 2012, and he organized the SRNTE, Society for Nicotine and Tobacco Research, in Helsinki in 2012. He's attending the SRNT annual meeting in Seattle in February 2014. He's also served as president of the International Society for Twin Studies. So please welcome Dr. Kaprio.
Okay. Thank you very much. As you notice, I have no disclosures, and it's a great pleasure to be here to give this talk to you. Indeed, I graduated from the medical school in Helsinki, then have been working in research and teaching since then. The topic of my talk is on the public health impact of rare variants and some of the issues and problems that we get when studying these. And as a bit of background, I just want to sort of give a brief history of where we are in this sort of field of genetic epidemiology and the genetics of complex disease, and then tell you something about Finnish population history, Finnish disease heritage, describe some of the large population cohorts that we have available for these studies, and then give a couple of examples from 2 studies that I've been involved in that are on a rare variant for schizophrenia and a rare variant for behavior impulsiveness. And then, if I have time at the end, summarize a project that we have initiated a short while ago called the Sequence Initiative Suomi, and some of the pilot work that we have under review for that. So I'll rather quickly go through what most of you all know about genome-wide association scans. After the sequencing of the human genome, we had the HapMap project to understand human genetic variation. There was advances in genotyping technology, so instead of doing a few SNPs at a time, then hundreds of SNPs, then tens of thousands, we can now do 500,000 to 1 million on a chip for quite a reasonable price. And the third essential component of this is the fact that you have had to have biobank sample collections of adequate size to address questions of interest. And realizing that capturing a fair proportion of the common variants in Europeans, you could capture much of the genetic variation by just genotyping 500,000 or 1 million of these. Then you find a case control series, you genotype them, and then you do simple 2x3 tables of the genotype by case control status, and then control for multiple testing. So you run essentially 500,000 tests or 1 million tests, and then see which of these become significant. Over the past year, since 2006, 2007, this has been an eminently successful enterprise. We have hundreds of genome-wide association studies that have been done. We have thousands of loci, and a few of them have been followed up now rigorously for functional information, and we have much insight into the biology that we didn't have earlier. In psychiatry and psychiatric conditions, there was a feeling for a long time that the model that was so successful for things like type 2 diabetes or height or body mass index wasn't really taking us anywhere. But as sample size increased, even for a disease as complex in its nature and phenotype as schizophrenia, we now have multiple loci, each of which is a unique biological signal to follow up on. Not all of them will be relevant, not all of them will tell us more about the pathophysiology. But for instance, in the October last year meeting of the World Congress on Psychiatric Genetics, the Psychiatric Genetics Consortium reported on finding 130 loci on a 38,000 GWAS case-control data set. So clearly progress even in these phenotypes that are considered to be quite intractable has been found. So what have we learned? We've learned that, first of all, very large sample sizes are needed to achieve genome-wide significance. The concept of sort of common disease, common variant, ones of large effect sizes, has really shifted to finding a more polygenic structure where there are multiple genes of small effects, typically odds ratios of 1.05, 1.1, and you need a very large sample size to find these. So the largest one that I know of at the moment on height under review in Nature has 350,000 people in it. It finds 650 loci for height. There are some 400 affiliated institutions involved in the study, I think some 450 to 500 co-authors on the paper. So it's a large, big science group effort that really rivals, I think, large-scale physics as well. And then you pick out those for more detailed studies. In the same way, the first large-scale study for smoking had 10,000 smokers from DCODE, the Icelandic sample, identifying the chromosome 15 alpha-5 variant that has then been shown to be functional and being followed up with a lot of very interesting mice and work. These have also taught us that we need to pay attention to signals that are not quite genome-wide significant. They can be informative. The method of doing this by combining samples from very many different countries, of course, can give rise to sample heterogeneity. And on the other hand, they've also taught us that if you improve the phenotypic accuracy, you can get by with much smaller sample sizes. So that for the chromosome 15 alpha-5 receptor variant, it explains about 1% of the amount in cigarettes smoked per day. So it's not a very large fraction of the variance that is accounted for. But then if you take a biomarker of nicotine intake, that is to say cotinine, which is a long half-life metabolite, then you are accounting already for 4 to 5% of the variance. And just last week, we reported here at the meeting of SRNT on a new GWAS of cotinine levels. With just 5,000 people, we were getting p-values and associations that had been shown for 100,000 people in the 2010 Nature Genetics set of smoking papers. Then, of course, technology has moved forward. We have better coverage of the genome, more accurate sequence data, and imputation first to HapMap and then 2000 genome data shows that you can find more signals in even the same data set when it is imputed more accurately. And then going to population-specific reference sets also improves SNP imputation. We've shown that in the Finnish population. Despite all these advances, we still have a substantial fraction of the family-based heritability estimated for family studies or twin studies that remains unaccounted for by common variants. And this is often called the missing or hidden heritability issue. And one of the proposed solutions for addressing that is then the study of rare variants, the ones that are not covered by these common variants taken as a minor allele frequency of 5 considerably. So the hype first was that finding common variants for complex disease, we would find sort of high penetrance common variants. These are rare. We have ApoE for Alzheimer's, and there are a handful of others, but most of them have very small, even vanishingly small effect sizes of themselves. Together, they typically explain 5 to 15% at most of the variance. But if you count them all together, all the common SNPs, they typically explain about half of the variance of the that is accounted for by heritability. So the interest now is going into these low frequency variants, and as you go into those, they come up with their own issues. And of course, there's a very nice paper sort of covering the issues of why study rare variants. I would refer you to that to find more details. So then, going on to the population history of Finland, this is on almost the other side of the globe. The time difference is 10 time zones. We are nestled there between Russia and Sweden, up Norway up north, Estonia to the south. And for a long time, this was quite isolated, both geographically. There was a cultural border between Western and Eastern Christianity between Finland and Russia. We were part of the Kingdom of Sweden until 1809. Then we became a Grand Duchy under the Russian Tsarist Empire, became independent of Russia when it became the Soviet Union, and fought them a couple of wars, maintaining our borders, and were never occupied during the Second World War. So that's how we have a unique situation in that sense. We have our own quite distinct language, not related to the mainstream European languages. That has been part of the isolation. Our population increased rapidly over the past few centuries, but essentially very little immigration. So still 20 years ago, less than 1% of the population was foreign-born. Now with joining the European Union, opening up the borders, we have had much more immigration. It's a much more mixed population, but still largely 97-98% Caucasian of European descent. We have our own genetics that I'll come back to. So Finland was settled when the ice mass retreated, probably some 10,000 years ago, but with the same population for the first thousands of years. And that was mainly situated along the coast. We had the early settlement areas, south and west coast. And it was only later in the 16th century when the King of Sweden wanted to have the inland parts populated to keep the Russians from coming from the east. And as a result of that movement, we have had then all these sort of minor border wars, conflicts, but a sparsely populated inland area has always been the case. As a result of that, the population in these late settlement areas are really the descendants of small groups of people that then expanded. As the population expanded, the food situation became better, and this resulted in both the Finnish population as a whole being a population isolate, but also we have internal isolates out in different areas. So if we have a bottleneck effect in a population, you have a famine, infections, so on, the population steadily reduces, and instead of having the full mix of genetic variation that you have in a total population, you have some chance survivors that represent a much smaller fraction of the genetic variation that exists. And that's the situation for Finland and our internal isolates. As a result of that, we have the so-called Finnish disease inheritage, where we have many rare diseases that are much more common in Finland or don't exist anywhere else. And on the other hand, there are diseases that are not found in Finland. We don't have cystic fibrosis, we don't have Huntington's. So any case of Huntington's is someone that's come late into Finland. But I'm not going to go to the clinical genetics, but just to show that in population simulation effects, there has been shown that we have had a bottleneck some 100 generations ago at least, and you can simulate that population structure to account for why we have a lot of these rare variants. Within Finland, we have also internal isolates. One of these is the Kuusamo region in northeastern Finland, originally founded by 40 families sent there by the king to inhabit that region. The northern parts of Finland here is approximately the size of Belgium or the Netherlands, so we're a rather big country. So at the moment, Helsinki, down south, the capital, has rain and no snow. Up in Lapland, we have a meter or more of snow. The increase to about 20,000 inhabitants by 1995 with negligible immigration. And that region has then become enriched for some common diseases as well. So schizophrenia is there about 3 times more common as is elsewhere. There's also an increase of asthma that is being found there. If one then looks at the genome-wide array-based relationships using principal components analyses of Europeans, the Europeans generally map quite well on the geographical distribution. We have the Italians in the south and the Icelanders up north and the Germans and Scottish and England in the middle, but Finland is way off. So the Finns are up here, and then you take the Finland isolate, Kuusamo region, it's even more distinct. So we have a very distinct population history in this respect. This also has permitted mapping of these disease genes with very, in early stage, work done by Reijo Norja, Leena Peltonen, and others have permitted mapping of these. And because we know our population history going back to genealogies back to 1640s on the church books, we have an unbroken series of parish records. So I know where my great-great-great-great-great-great-grandfather was born in the southernmost corner here. He was an officer in the Swedish Army and served as the captain of a castle south of St. Petersburg called the Kaprio Castle, which then my parents, my father and his siblings, took the family surname when we changed from a Swedish to a Finnish-speaking surname. The uniqueness of these rare diseases, of course, quite well-known history. Now we're trying to see whether we can use this population structure to our advantage when we study common diseases and using Finnish population cohorts. We have had, of course, many individual studies in Finland run by the university research groups or research groups at the National Institute for Health and Welfare. The FINRIS studies are every 5 years, and we've been collecting DNA on these subjects since 1992. These were primarily a public health monitoring exercise. Health 2000 was also a public health examination with about 10,000 people. The Young Finns cohort has been a study of children and adolescents. In our twin cohort, we have about 70,000 individuals that I'm responsible for. The North Finland birth cohorts are 2 birth cohorts born in 1966, 1986 from northern Finland. And there are many others. So we have the possibility of using nationwide medical registers to find out and follow up on medical events. So these, of course, cause the sort of cause of death register, the hospital discharge register. There's a drug reimbursement register for every single prescription that is used in Finland since 1995, a nationwide cancer register, and with appropriate permissions and consents, you then can link these to the population cohorts. As a result of also then active genome-wide association studies, we've been participating in multiple gene discoveries for many conditions overall. And we have then compiled these into a database of the National Biobanks Finland. There's about 130,000 individuals from these population cohorts and 70,000 individuals from disease collections of different types. And this is all made possible by the fact that we do have a unique identification number for each Finnish citizen, assigned first to all Finnish citizens in 1960s, and then now currently at birth. So it's your date of birth, then a 3-digit number that is paired for women and odd for men. So you get the gender, sex out of that immediately, and then a check code. And that is used in all these medical registers, as well as all official registers otherwise by the government and local authorities, but also widely by some of the sort of largest companies in Finland as well, if you consent to give that. We're much more trusting of our government than I believe that the U.S. People are. And there's not been, to our knowledge, a single breach of security or confidentiality from these medical studies. The banking side is a story of its own. Not going to that. So now we're going into this sort of low frequency variants. Can we find more information about disease predisposing alleles for common diseases? We have it for the rare ones, obviously, and the idea then is that if we go to common alleles, is there really a true bottleneck effect, or were these sufficiently common that even the small group of founders, there would be enough variation for them to be retained, and that remains to be shown. Because of the relatedness in the longer LD blocks that we have in Finland, it's been shown that we can, in fact, with reasonable sample sizes, reconstruct parental haplotypes and really create a population-based genome structure. For small island populations and internal, you get up to those sufficient numbers quite quickly, but even for the whole of Finland, it's sort of reasonable with 5,000 samples or more that have been whole genome-wide sequenced. For something like the UK, it's a virtually impossible task. So what has been done so far in most studies is that you take a phenotype, you take people with, say, diabetes and controls, you genotype them and infer the genotypes that are predisposing to the disease. You get some idea of the genetic loading, but you most of all get ideas on what are the population biological effects that you're looking for. So for type 2 diabetes, we've learned a lot about beta cell function and where the genesis of diabetes really probably comes from. But we're getting to a position where we can also begin to look by genotype to see what are all the phenotypes that are related to that genotype. So it's a two-way street, and the studies are based on genotype, of course, are rarer than those based on phenotype. One example, we go back to the Kuusamo population, and there was a case control study of, within the cohort of Kuusamo, of 174 schizophrenia patients, and then unaffected controls that had been assessed psychiatrically. And this is work that was reported last year by Stoll and Pietiläinen in Nature Neuroscience, and I will refer you to that for more details. They were looking at CNVs, copy number variants, to see whether there's an association between the disease and these loci, and found a locus on chromosome 22 that showed modest association with schizophrenia. There was also 4 individuals with homozygous deletion. They were distantly related and lived or came from the Kuusamo and related areas. Then this was genotyped on other samples in the isolate, showing an odds ratio of 1.84, and then in many of our population cohorts, including the twins, where we find then a odds ratio of 2.6, so it's in fact a stronger effect than that isolate, and highly significant, partly due to the large sample sizes. And then in the International Schizophrenia Consortium and a Swedish sample with very large sample size, didn't show an association, but combined still retained. So this is more or less a Finnish-specific variant. The next step was then to see whether this has other effects then on schizophrenia, and looking at in the North Finnish birth cohort, where they had many phenotypes that were relevant on mental retardation, intellectual deficiency, and the simple measure of whether you had repeated grades at school showed an association with this variant. So the variant carriers had repeated more than others, but no association with a number of other mental and central nervous system conditions. And this deleted fragment that comes out of this CNV was then contains the topoisomerase 3 beta gene. It's functionally involved in the Fragile X mental retardation protein, and so nicely shows a functional work. And there's a big chunk of functional work in that paper that I will not go into here. It's nothing that relevant. But really the fact that you get a finding and then you can see what is it associated with a specific phenotype, does it have, which would then implicate its causality much more than if it was sort of widespread over different types of phenotypes. And just showing the value of getting rather crude even phenotypes, something like being repeating grades at school, which is exceptional in the Finnish schooling system. So it does really indicate that there's something serious going on there. The other example I'll go to is one that I was involved in. We published with David Goldman in Nature a few years ago, where we identified a gene that was possibly predisposing to violence while intoxicated. Finnish drinking habits used to be quite, and still are on time, quite binge drinking weighted, and this is probably a phenotype that's less obvious elsewhere. So we were looking for traits that were associated with impulsivity in the sense of associated with major psychiatric conditions such as suicidality, impulsive criminal behavior, harming oneself or others while intoxicated, doing things that one would not do otherwise. And it continues to be, of course, quite challenging to find variants associated with psychiatric and psychological traits. We've done a large-scale GWAS of some personality traits such as extroversion, neuroticism. Even with 100,000 people, we don't find associations. So this was a targeted exome sequencing of severely impulsive individuals that were treated in a psychiatric criminal ward, assessed by Matti Virkkunen, and 14 genes belonging to the serotonin and dopamine system were targeted for 96 cases. This was sort of work done in 2009, and controls. And then out of the variants that were then assessed there was some assessment of their functionality. There were then genotyped, the ones that we took forward in all cases of controls, and tested for case control differences. One of the ones that came out, in fact, the only one that came out was a functional stop codon in the serotonin 2B receptor, which should be common in this population sample with a frequency of 1.1%. And that was associated with the psychiatric case status, and especially in those that were marked by impulsive behaviors. So people who had been drinking and suddenly took out a knife and killed someone. That was the type of cases, and in fact it's found in all the repeat homicide cases in this series. It co-segregated in 2 generations in 8 families where there was this same severe psychiatric morbidity in both parents or one of the parents and the offspring. And this fraction is then higher than we would expect. We genotyped almost 5,000 controls, finding 106 heterozygotes, 1 homozygote, and the allele frequency then was in the population 1.1%. David Goldman screened for these in the U.S. among almost 600 cases and 2,500 controls. One carrier was found, happened to be of Finnish descent. So seems to be a Finland-specific mutation that is not found elsewhere, and just indicating that as you go to rare variants, you'll be finding more and more ones that are population-specific, and then finding the correct reference population will be much more of a challenge than any mixed population. Can you really use the general population frequencies as the correct reference values when you're computing risk? And that just shows where the stop codon was situated. We had in the Finnish twin cohorts detailed neurocognitive measures, and we found that the heterozygote men, but not the women, had significantly worse working memory on the digit span test. Otherwise, they were normally cognitive, and their sort of overall behavioral information from questionnaires, interviews was otherwise normal. But they seem to have a selective deficit in frontal cognitive function. And there was a bunch of mouse work that sort of backed this up. We further genotyped these. As I mentioned, the carriers seem to be overall socially and cognitively normal. The one homozygote was also had nothing special about him. Well, even though we had this detailed cognitive information, the number of carriers was limited, so we have now sought to extend that to other samples. These were young adults, to see whether we see any effects of being a carrier in older samples. Or does it, being a carrier by itself, is it just simply neutral, and you really need other genetic predisposing or environmental factors to bring out that impulsive behavior? So, I mean, you can compute that about 100,000 Finns out of a population of 5 million are carrying the variant, yet only a minor fraction of them exhibit the extreme phenotype of impulsive, violent behavior. And either it's not seen in the population, or then there are these other triggers. So we genotyped in the FinRisk study, which is a population-based study of about 10,000 individuals every 5 years. We had 26,000 people with DNA that could be successfully genotyped for the stop codon. There we're already getting up to 6 homozygotes and 904 heterozygotes. The allele frequency, in fact, was slightly larger than in the initial sample, nicely in Hardy-Weinberg. No association with sex and age, but all but one of the homozygotes were men. And if you add in that one original homozygote, we are sort of pushing the borders of a significant gender difference. We found that the variant was somewhat more common in those living in northern Finland. You might speculate that up north, where it's less populated, you probably have to have some more different impulsive characteristics to survive than further south. But of course, that's just speculation. Looking at sort of things that could be consequent to impulsive behavior, we found that the male variant carriers were less often single and more often divorced at a sort of very marginal level of significance. Direction of effect was the same in women, but not significant. And my explanation here, that if this was true, it could be that the impulsive ones are the ones that decided to get married, quite possibly. They proposed to their spouses, but then because they're impulsive, they also then divorce more often. It's just one possible explanation. We don't know that without studying them in much more detail. We found a significant association with the educational group, such that the carriers were more often in the lowest third of educational years, which was a standard classification used by that population study. Given that they were studied every 5 years and educational level has been increasing, we had to do a standardization of the sort of within survey educational level to achieve that. That was more significant. But on the other hand, we were already testing a number of phenotypes, and sort of this is on the border of surviving multiple testing, and no association in the women. Looking at a number of other phenotypes, no difference for a whole host of diseases, being on disability pension, self-rated health, so no association with drinking frequency or intoxication. They were marginally more often current smokers, which would tie in with some of the literature on impulsivity and smoking initiation, but not depression or other medication use. So this sort of was promising that you could see some impact of being a rare variant carrier in the population, but still very cautious about making conclusions. And you could say that the lower education in men is consistent with the correlates and consequences of impulsivity. This actual variant was not significant in the education multi-site GWAS analysis that was published in Science last year, to which there was a number of Finnish contributions. Because it's not found anywhere else, then it simply washes out of any large GWAS study. So that in order to study something like this or find them, you would have to do within the population of interest very large GWAS studies. So that would really require quite substantial resources, and we'll go into how we're trying to address that issue. Then we did another smaller replication data set, the Health 2000, which only had 4,500 people, finding 113 heterozygotes, but absolutely no sign of any association with education, immersion status, or smoking that we found there. So that's the sort of current situation we are with this study. We haven't published this. The next round is to go to the other data sets we have in Finland that I showed you and see whether we retain any findings or not, and then we can make some conclusions about whether this variant has any impact on any other phenotype, or is it something that we see only when combined with special circumstances that these impulsive criminals have, and what those are need to be done. And we're planning a study of studying just the actual variant carriers once we get that through ethics. And then the concluding part of what I want to share with you comes from the SISU project. This is a sequencing initiative Suomi project where we're collaborating a lot with people both in Sweden, the UK, and the United States. Over the years, we now have a total of about 60,000 GWASs that have been done in Finland. By the end of this year, that will all be available. The last 10,000 are sort of in the pipeline. And then there are sort of whole genome or exome sequences on 30,000 people. And that is giving us a database that will enable us to essentially impute all the existing GWASs to high accuracy and generate information on variants that are specific to Finland. And then the goal would be to do population-specific genotyping on a chip or otherwise of all the samples that have not yet been genotyped, and we can look at things then that are specific to Finland, but on the other hand, their biology would be universal. As the first pass, we'll be looking at some loss-of-function variants that may, of course, be potential shortcuts for drug targets. And for the first study that we have then compared 3,000 exome sequences from Finns with 3,000 exomes for non-Finn Europeans. Sorry for the typo there, there's a missing 3, so 3,000 and 3,000. And if we compare the relative frequencies of different variants at different allele frequencies in Finns versus the non-Finns, so this is the ratio of the expected frequency in Finns compared to non-Finns, we see that there is a excess of variance in the sort of 1 to 5% range and 0.5% to 1% range, particularly of loss of function variance and a underrepresentation of the less frequent ones, the ones that are very individual or singletons, doubletons, compared to European ones. So extreme rare variation is depleted in the Finnish ones, so indicating that most of the rare variants do not make it sort of through the bottleneck and have not survived. Of course, there have been then recent mutations occasionally after that as well. Surviving rare variants seem to get a big frequency boost as the population has expanded, and some of these boosted variants are more damaging as seen by our disease heritage. Are these visible when you look at common traits or not is then an interesting question. So then all the single nucleotide variants and indels from this frequency range were taken forward, 103 variants, and genotyped in the FinRisk and Health 2000 sample, about 35,000 people. And as a first pass, we just looked at existing register and biochemical information that could seem to be medically relevant. And so there are a whole host of biochemical measures and physiological ones, sort of all the lipids, blood pressure, etc. And then these have been linked to population register diseases. These were classified. There was no associations with the actual diseases. They were too rare, but finding associations with reduced lipoprotein A levels, galactin, triglyceride levels, and D-dimer levels. And we're at the moment now, at the request of the reviewers of the paper, replicating and extending those association studies to some new samples. It's been difficult to identify these because we have to work with Finnish samples. We can't go to any other European samples to do this because these are Finnish-specific samples, variants for the most part. So we see, as you well know, there's a strong association of LPA levels with coronary heart disease risk, and we find that this loss of function variant is enriched in Finland. It's tenfold more common than elsewhere in Europe. And there's strong association with LPA levels and also with CHD risk in the carriers than compared to controls. So the sort of take-home message there is sort of unique potential for discovery in this population. We have a sort of very well characterized population for the genomic studies, and combined with 40-plus years of registry follow-up for health events. So our medical registries that have been computerized were all initiated in the late 60s, early 70s. The ideal then would be to use these to do sort of personalized or stratified medicine for risks, and we have just initiated a pilot study among blood donors. We asking them, do you want to be genotyped for a panel of 100 SNPs that are associated with cardiovascular risk that do not seem to go through the traditional risk factors, and seeing whether the population accepts this and wants to be interested in that. And there's been a fair bit of media attention within the Finnish society for that. So just to conclude, I would say that genomic data will be part of the basis for personalized or better stratified medicine. We are certainly in the very early stages of doing that. We have some nice examples from especially cancer and infectious diseases, but for other common diseases, we're still much more less advanced. There is meager inclusion of relevant environmental data, even simple things like physical activity, smoking, alcohol use, that we know to be relevant. And I would still say that overall the gene discoveries that we get out of these studies are more valuable as insights into mechanisms of pathology than in terms of targeting individuals at risk or identifying people who require different therapies. So with that, I would like to conclude this talk, and I'll be happy to take any questions.
The fact that people are remarkably trusting about having this amazing database so you can cross-link all these different things. And the fact that you have this very kind of bottleneck population, do you have any idea of how immigration is going to affect your ability to keep doing this sort of thing?
I mean, of course, we have some already ethnic minorities like the Sami in northern Finland. Then we have a linguistic minority, the Swedish-speaking ones, but their genetic makeup is essentially the same as Finns. So that language choice that you make, or your parents and grandparents have made, is not really affecting. So, of course, our new population, immigrants, we have people from Kurds, Iraq, Syria, Somalia, Vietnam, so on now, will, of course, over time, make us more of a mixed population. But of course, we can still, at this stage, we have the population history, we have the samples and the disease history that we can use now to take it forward over the next 5, 10, 20 years. And then we have the same issues about declining participation rates in studies, so that you see elsewhere similar challenges in that respect. On the other hand, we have a new biobank law that clarifies some of the use of the samples in a more explicit way, a more systematic collection of consent for the use of samples, and so fully integrating into a European network for the protection of patient rights and so on. So that we try to tackle all of these and do a fair bit of education of the public about what its interest is, and then we have the sort of some competition from commercial gene testing units that need to be also considered in terms of either commercializing what we find, but also people selling products that do not necessarily apply to the Finnish population, sort of the 23andMe type of issues.
One more quick question. Since you have this database and you're actually it looks like able to link whether or not people get cancer and are smokers with their genetic data, is there anyone looking at, we're always curious why people can smoke 50 packs over their lifetime and not get COPD, not get cancer, and some people do. Is there anyone looking for genetic predispositions for that kind of thing?
Well, I gave a talk at the EPI seminar here last week, and there I talked about our Nordic twin cancer study, where we have 300,000 twins in the 4 Nordic countries, 40,000 cancer cases, and I'll be just writing up a paper on the heritability of lung cancer stratified by smoking status. So essentially, in non-smokers, we have 1 concordant pair, 100 discordant pairs where there's only the other twin has lung cancer. And you really can't say anything about the genetics of lung cancer in non-smokers. And the heritability overall is about 20%, which is consistent with both U.S. and earlier Nordic twin studies. Then you go to the people who were current smokers at baseline, the heritability goes up to about 50%. So there are genes that are probably evoked by the fact that you're a smoker. And we see that from the lung cancer GWAS studies as well, that there are lung cancer genes found for smokers and non-smokers. But then there are ones that are only associated in the smokers, notably the chromosome 15 alpha-5 nicotinic receptor B398N variant that seems to increase risk, for lung cancer if you're a smoker, but no association at all in several large studies that have come out from Hunt in Norway and other ones, showing absolutely no association in the non-smokers with lung cancer. So by itself, it doesn't induce lung cancer, but it requires the smoking to do that. Whether or not it has an independent role of smoking, or is it just a better indicator of smoking than our smoking history, remains to be shown. I think the jury is out on that. But certainly, these are highly interesting issues and things that we're following up on.
I'm curious if there's been any use of the evidence about impulsivity, alcohol drinking, as a legal defense in your court system. So if you've found that someone carries a gene, can this be used to defend their behavior?
I haven't heard that being used. Of course, we have a perhaps somewhat different legal system. We don't have a common law sort of Anglo-Saxon legal system, but a more Latin Roman codified system, and I think that it's less adversarial. So I haven't seen, I don't recall that genetic data has been used in either case for or against the defendant. But it has been considered as a basis for whether to grant a shortened prison sentence or not. What is the risk of reoccurrence? And the sort of criminal justice system and the forensic psychiatrists have discussed about whether should they and could they genotype people who have had impulsive criminal behavior, be sentenced to prison, say for, we have a life term that's essentially 12 years, and you can get out a bit earlier on good behavior. But then if you're deemed to be at high risk, then it can be renewed, so you don't get out after all. And so some of our worst pedophiles have been kept in prison, essentially lifetime. But the genetic data, to my knowledge, has not been used for that purpose. But I mean, we had we had one of these impulsive killers who was released after a full prison sentence, and the day after he was released, he went drinking and then killed at random 2 people in a car. So that might have been a case where you might have thought that would have been of usefulness. On the other hand, the variant doesn't explain all of that. It just explains a fraction of those. So you have people with impulsive criminal behavior that are not gene carriers. And what the predictive value is has not been yet shown. But it's an interesting question that will probably come up as more and more genotyping is available for all kinds of matters in sort of the forensic use of this data. But it's not, the police cannot access these databases.
Has anyone offered a hypothesis on how that the variant that you've identified in highly impulsive Finnish criminals might generate impulsivity?
It affects certainly serotonin levels in the mice models, and we have some ongoing studies to study the carriers in more detail, but I can't share that with you.
Okay, thanks a lot, everyone.